Biotechnology is now an essential field and rapidly advances as humanity advances. The discovery and usage of CRISPR especially has accelerated research on diseases. CRISPR, or clustered regularly interspaced short palindromic repeats, is a genome editing technology that allows us to modify, delete, and correct regions of DNA. With such technology, we can now genetically modify human embryos to relieve them of inherited diseases. This sounds delightful in theory. However, as with a lot of biotechnology, CRISPR is much more complex than just a miracle solution. There are regulations and ethical concerns to consider, which we will soon discuss. First discovered in 1987, CRISPR was perceived as repeat DNA sequences in prokaryotic organisms, which could likely be adaptive to the immune system. Since then, further discoveries and experiments in adaptive immunity, CRISPR in relation to RNA and DNA, and the Cas9 protein associated with CRISPR were developed. Finally, in 2013, CRISPR-Cas9 was harnessed for genome editing, and since then, the scientific world has had varying contributions and opinions on it.
The actual mechanism is complicated, but here's the breakdown. Starting with bacteria, an invader first injects genetic material which integrates into DNA. Transcription produces pre-CRRNA, which is enzymatically processed to become mature CRRNA. It's analogous to how the primary transcript becomes mRNA in eukaryotes. Transcription also produces tracker RNA from host DNA. The endonuclease that cleaves DNA, Cas9, is also translated from the Cas operon. In human methods, these steps are skipped. A hybrid manufactured sgRNA combines tracker RNA and CRRNA. It acts like a guide RNA to direct Cas9 to a complementary DNA sequence, similar to how a sigma factor binds and acts like a landing pad for RNA polymerase. The pre-made Cas9 enzyme is also inserted directly into the cell. In humans, frameshift mutations in the UIS gene can cause retinitis pigmentosa, a genetically inherited disease causing blindness. Application of CRISPR research in human embryos, although controversial, aims to cure this using a site-specific sgRNA to get Cas9 to cut and delete 5 base pairs from the gene, restoring the correct reading frame. Cas9 then joins the two ends, restoring the UIS gene to normal function.
There are currently many laws and regulations pertaining to gene editing research. Due to the ethical debates, CRISPR technology and its specific application to the human genome is regulated differently from country to country. There are currently no nations that allow modified germline cell implantation. There are, however, different legal and permitted standards for research on gene editing in embryonic cells. If we look at this table from the Global Gene Editing Regulation Tracker, we can see how stem cell regulation in the left column and human genome editing regulations in the right column vary greatly from country to country. Using Canada as a case study, the intricacies of research regulating policy become apparent. Certain regulations include: embryonic gene editing is prohibited under the Assisted Human Reproductive Act. Any modifications to the human genome or heritable traits is illegal and punishable by up to 10 years in prison. Non-human embryos such as mice or sheep embryos can undergo gene editing. Adult genes in the form of stem cells may be edited as long as the gene is not heritable. This is a stark contrast to countries like France and Sweden, which allow for human germline cell editing as long as the embryo is not used in pregnancy.
When CRISPR was discovered, it immediately became of great interest as it had the potential to be utilized in the prevention and treatment of human diseases. However, when the concept of applying this technology to germline cells came about, it brought up a number of ethical challenges. Nevertheless, in support of germline editing to eliminate genetic diseases, we can mention the following. Scientists agree that in some cases, germline editing can address needs that cannot be met by other medical methods. Thus, germline editing can ultimately offer the hope of editing cruel genetic mutations out of the gene pool. And where the concept of informed consent for germline therapy is concerned, as the patients affected by the edits will be the embryo and future generations, we need to acknowledge the fact that parents already possess the ability to make decisions that directly affect their children. Thus, the question then becomes, where do we draw the line between disease treatment and enhancement, and how do we enforce it? On which this quote by Robert True perfectly summarizes the key points.
As miraculous as the benefits of CRISPR appear to be, there comes an ethical cost to its wonders, the first being its relation to the concept of eugenics. CRISPR's ability to transition disabilities out of society may eventually lead to a full-scale movement of velvet eugenics, a subtle method of urging for the termination of disability, virtually masking it as a form of full-blown eugenics. By making genetic customization acceptable through velvet eugenics, CRISPR could propel the gene editing conversation into non-therapeutic realms, such as the selection of physical features like skin tone. Another problem with CRISPR? It isn't perfect. Although the editing of somatic cells has a lower risk of mutation, germline editing using CRISPR is significantly more prone to unintentional side effects caused by mutation. These mutations may negatively affect the health of future generations through being passed down, so many scientists warn against the immediate endorsement of CRISPR gene editing. CRISPR is a relatively new technology, and there will likely continue to be new developments for its application. Perhaps one day it will become the solution to inherited diseases, but for now, it has a long way to go.